Once you have prepared your sample from your cheek, your group will have labeled PCR tubes. We're going to extract the DNA from your cheek cells, and we're going to use the PCR machine to do the heating step. So in order to do the heating step, make sure that your lid, you have rotated this knob all the way counterclockwise. You open the lid. You place your samples. There's a place inside for you to place your samples. You might have up to 6 if you have 6 people in your group, and then you close it until you hear it click. To tighten the lid, you take the knob on the top and you rotate it clockwise until it's finger tight. The next step is going to be to make sure that the machine is plugged in. So there is a power cord that you will need to put into the PCR machine on one end and plug the other end of the plug into the outlet at your lab bench. You will also have to plug in the USB cord. One plug will be in your PCR machine. The other is going to be in your laptop. Once you've done that, there will be a place for you to turn this on. There is a rocker switch. Turn it on. And now you're ready to run your protocol. In order to run your PCR program, in this case we're going to do the DNA extraction protocol, which you have already loaded into the Mini PCR software. So it should be under your library. Here's the PTR DNA extraction protocol that you've typed in previously. And now you are going to go over to the triangle that says Run and simply press Run. When you do that, you'll see the word loading. And if you've properly connected your device, it should be loading the software into the PCR. And then it tells you what it's beginning to do. So in this case, it is loaded. It says Now Running, and it is heating the lid. On the screen, it'll show you what the temperature is. So right now, it's showing that it is at 60 degrees. And what you will see is as the lid heats, you'll see this begin to rise. And then it will actually start the heat step, in this example, at 95 degrees for 10 minutes. All right, so we are now at the end of the run, and it marks finished. And you can see this line that has gone through all the way to the end. When the run is finished, then you can take out your tubes. By pinching between your thumb and forefinger on the side of the box, carefully opening up the tube, and then removing your PCR tubes. They will be hot on the bottom, so just touch them on the top and place them in your rack. So each of you will have a sample now of your extracted DNA, and you're going to be using that DNA extract from your cheek along with EZ-PCR Master Mix and PTC Primer Mix for the next part of the experiment. So follow your protocol to make your tubes. And the next step is going to be to perform the PCR reaction on those newly prepared tubes.